Hi there, what's up? Today we are on Beyond Genetics, where advice no returns, sub-page. So let's get started. Oh no, are we diving in again?
Yeah, hello friends, I'm your host today, and before you ask, no, I'm not in a fishbowl. But technically yes, but it's an existential metaphor. My name is hard to pronounce, but my friends call me Tiki. I'm Tiktaalik, not TikTok. And yeah, I was the first to take the giant leap from water to land 375 million years ago. Why am I here? Because if anyone gets successful evolutionary screw-ups, it's me.
Hey, me too. Don't interrupt me, alright? As I was saying, I've got things that I always learned, yields and primitive ones. I'm basically nature's most successful copy-paste hero. I wouldn't exactly say successful. Well, maybe after we record this podcast. Anyway. Today, we are talking about when life's instruction manual, our DNA, comes with typos. And trust me, I've seen lots of typos in a million of years. Let us start with Charles II of Spain, the king born in 1661. For painters how to be creative. His jaw was so prominent he couldn't shape properly. The reason? His family tree was more like a family circle. His parents were uncle and niece, his grandparents were cousins, and if you keep going up, well, let's just say family reunions were in. So many marriages between close relatives concentrated genetic mutations like dropping all your marbles in the same hole. Hey, in my day, we were all distant cousins, but at least we had a whole ocean to mix things up. Jump to the 20th century with André René Rosimond, better known as André the Giant. He was 7 feet 4 inches and weighed 520 pounds. It wasn't magic or radiation, it was acromegaly, caused by a tumor in his pituitary gland. It produced growth hormone like a factory with no pump but his chromosome didn't have a copy error. The soldiers decided to go rogue and go out of country. And my third historical suggestion, Rosemary Kennedy, KF his sister, born in 1918 with high intellectual disability, possibly from lack of oxygen at birth. But here's where it gets interesting. Her story shows how society has treated genetic and neurological difference. They lobotomized her at 23 to calm her down. It wasn't a genetic problem, but it teaches us that sometimes the real error isn't in the chromosomes, but in how we respond to difference.
Yeah, but at least explain what a chromosome is first, right?
Sure, think of chromosomes like instruction books, a strand of chromatin filament that contains DNA. Humans have 46 books, organized in 23 pairs. A karyotype is like taking a photo of all these books lined up in the library. And I see some karyotype examples here. The first is a typical male. We've got 22 pairs of autosomal chromosomes. The ones that don't determine sex, plus one X and one Y chromosome. The Y is the tiny one, like that thin pottery book among encyclopedias. It contains just 55 million genetic letters, while the X has 155 million.
In my time, we didn't have such defined sex chromosomes. Some fish change sex based on temperature. You mammals really complicate things.
The second karyotype is a typical female. Same deal, with the two X chromosomes. Here's a fascinating trick. Female cells randomly turn off one X in each cell. That's why calico cats have patches. Each patch is a group of cells that turn off a different X.
Exactly. And then come the better known typos, like Down syndrome. There's an extra book here.
3 copies of chromosomes 21 instead of 2.
It's like the printer got stuck and repeated a chapter. The genes aren't wrong, there's just too many cooks in the genetic kitchen.
Or Turner syndrome. Here, a whole book is missing. Just one X chromosome. It's the only case where you can survive missing an entire chromosome. People with Turner are women, usually short, and may have trouble with spatial math, but often have a superior verbal skills.
But why human? That's just the tip of the genetic iceberg. Those you mentioned are numerical alterations, but the level of errors is way more extensive and fascinating. It's the source of evolution, for better or worse.
You're right. Actually, I've got an exhaustive classification here. Let's start with us, eukaryotes, or eukaryotes. Numerical alterations split into two. First is eunoploidy, which is exactly what we described, losing or gaining individual chromosomes, like in Down or Turner syndrome. The cause is usually non-disjunction, a failure to split chromosomes properly when making eggs or sperm.
And second is euploidy, my favorite. It's not gaining or losing one chromosome, but a whole set. Mine having tripled the books, triploidy. Or quadruple,
Tetraploidy.
in animals, that's almost always lethal, but in plants, that's where the party is at. That's how new species are created, bigger fruits, short flowers. Nature experimenting big time.
Okay, that covers quantity errors, but then there are structural alterations. Here the number of books is right, but they are broken, badly bound, or have swapped chapters. Like deletion, losing a chunk of chromosome. It's like ripping out a key chapter. The consequences can be serious, like in Cri2-Chat syndrome.
Then there's the opposite, duplication, evolution's copy-paste. A chromosome segment appears twice. This is super important. You get a backup copy of the gene, so the new one can experiment, mutate, and suddenly you get a thing that starts looking like a leg. It's a source of new genes and functions.
There are also inversion. A piece of chromosome breaks, flips 120 degrees and reattaches. All the letters are there, but in the wrong order. This can cause huge problems when creating gametes and reduce fertility.
What a mess! And what about translocation? Now that's an organizational disaster.
Totally. Translocation is when a piece of one chromosome breaks off and sticks to another chromosome. That's not inspiring. It's like a chapter from the history encyclopedia ending up in the cookbook. This is behind some cancers like chronic myeloid leukemia and can also cause infertility.
An insertion is similar, right? A fragment from one chromosome inserts into another. A chapter from another book forced into yours. Chaos.
Exactly. And to find with eukaryotes, there are two rare but incredible cases. Isochromosome, which is a chromosome with two identical arms for an error in centromer division, and ring chromosome, formed when a chromosome loses its tips and the broken ends fuse, forming a circle.
Wow! You eukaryotes and your tricks. In my day, things were simple. Prokaryote life, bacteria and archaea have their own repertoire of errors. Since we usually have just one circular chromosome, numerical alterations are super rare. But structurally, that's where the action is.
Tell me, wise fish, how do bacteria mess up?
We also have deletions, duplications, and inversions. They're a bit of us. A duplication can get at redundancy, and from there,
evolve new functions. But the real specialty is insertion. It's our superpower, not used for errors, but because we incorporate DNA from outside, from plasmids, from viruses. That's how we pass around antibiotic resistance, like trading cards addresses. It's called horizontal gene transfer. So for you guys, insertion is a survival strategy. The best there is. We also do translocation, moving genes between our main chromosome and those little DNA rings called plasmids. And don't forget, gene amplification. If an antibiotic attacks us, sometimes we respond by making hundreds of copies of the gene that gives us resistance. It's an arms race at the microscopic level. Fascinating. So the general causes of all these errors are similar. Failures in copying DNA, errors in dividing chromosomes, external agents like chemical or radiation. But the consequences and frequency are very different. Totally. Unipolarity is your drama, but for us it's almost non-existent. Horizontal transfer is our daily bread, while for you it's rarer. In the end, all of this, from cancer to antibiotic resistance, from genetic disease to plant speciation, is the same story: genetic variation. The genetic crystal ball dilemma. And here, Tiki, this is where things get thorny. Because now we can look at these karyotypes before a baby is born. It's like having a trailer for a life that hasn't started yet. But what do we do with that information? Look, I was the first fish to walk. If my parents had seen my karyotype and said, "Fins with fingers," what an abomination! You wouldn't be here. Sometimes errors are just different paths. In Iceland, almost 100% of pregnancies diagnosed with Down syndrome are terminated. In other words, in other countries, rates vary enormously. Is this modern eugenics or personal choice? Where do we draw the line between preventing suffering and eliminating diversity? And when the test is wrong, false positives exist. Imagine making an irreversible decision based on a lab error. The future is in the genes, or is it? Technology advanced. Now we can detect not just extra or missing chromosomes, but specific mutations. We can know if someone has a predisposition to Alzheimer's, breast cancer, diabetes, etc. But do we want to know at 18, at birth, before birth? In my time, natural selection was simple. Either you learned to breathe air or you didn't. Now you can choose. It's a tremendous superpower, and tremendous is the way of using it wisely. Some countries ban genetic testing for sex determination because they know it would lead to selective abortions. Others allow selecting embryos free from serious genetic disease. Where's the limit? Fatal disease? Disabilities? Predispositions? Hate? Eye color? Closing the chromosomal circle. So, what did we learn today? That chromosomal errors in both eukaryotes and prokaryotes are a fundamental source of genetic variability and therefore evolution. Shows taught us that forcing genetic purity is a dead end. And dead again show us that even extreme difference can find their place. And Rosemary Kennedy reminded us that the real problem often isn't genetics, but our response to it. Look, I've been on this planet for 375 million years, in one form or another. I've seen perfectly normal species go extinct and evolutionary errors thrive. My old fish advice: diversity isn't a bug in the system, it is the system. Most of these errors are harmful, but some can confer adaptive advantages. Every karyotype tells a story. Some are tragedies, others are adventures. Many are used differently, and that's okay. After all, if a fish with legs could walk, who are we to say what's normal? Genetics doesn't come with returns, that's true. But it also doesn't come with a single manual for happiness. Sometimes the typos write the most interesting stories. I'm Tiki, the Tiktaalik, reminding you that we are all successful mutants in some way. Until next time, and remember, keep your fins wet and your minds open.